Thank you, Billy, for this nice introduction, and thank you, everybody, for the invitation. I'm very honored for this opportunity to present my work and also the work of many collaborators in the team. Before I start, sorry. So nowadays, I'm working in Wilfried Thuil's lab as a postdoctoral research fellow. You probably know the Alpine Ecology Lab. The team works on both research aiming to understand the mechanisms behind biodiversity patterns, but also in predicting them in space. And it is located in the beautiful city of Grenoble that is surrounded by mountains. And for me, who comes from a mountainous area in the Georgia National Park in northern Algeria, it is like being home, away from home. So that is very nice. Coming here was a succession of serendipitous events that started with first my training in computer science, and then my PhD in ecology, where I met this very nice team of researchers from both ecology and computer science. So Michael Ed is a soil ecologist from INRAE in Montpellier. Wilfried Thuil is an ecological modeler and ecologist from CNRS, and Esther Galbran is a computer scientist. And I was very lucky to do my PhD under their supervision, where I worked on deep learning for joint species distribution modeling and also on inference of ecological networks. After my PhD, I did a postdoc in the Team Lab, which is also located in Grenoble, with Olivier François, where I worked on environmental DNA. And the first part of my talk will be about the work that is related to that postdoc. And then since 2022, I joined LECA officially as a research fellow, where I work on various methodological developments related to AI for biodiversity modeling, with applications in various projects, and with also many empirical applications supporting conservation projects such as the Natura Connect project in Europe or the Obscussion project. During this talk, I will just provide a general context that is information that you all already know, but at least it gives time for people to join a bit late. And then in the first part, I will talk about how we account for ecological interaction networks in biodiversity models. And then in the second part, how we can use AI to better characterize and describe landscapes from Earth observation data for biodiversity forecasting. For each of these, what I refer to as modeling challenges, I will provide some motivation, describe the methods that we suggest, illustrated through a case study, and then at the end discuss the implications for forecasting and also some practical considerations. And I will end with a summary. So let's start with a bit of context. So you all know this is not new information, but it never hurts to remind ourselves of that. More than a quarter of species on average assessed by the EUCN are at risk of extinction, and many more are not assessed. So this is just the tip of the iceberg. This loss of biodiversity has been attributed to different causes, including climate change, but not only that, also land use change, habitat destruction, direct exploitation of organisms, pollution, and invasive alien species. And due to this pressure, there is a need for ecosystem management actions that include conservation to protect biodiversity from anthropogenic pressures or to mitigate the effects of climate change, but also restoration. So all the processes that are needed to restore degraded habitats for biodiversity or to reintroduce species to their natural habitats. To support these management actions, the team where I'm working in Wilfried Thuil's lab, we are specialized in developing biodiversity models. And for this presentation, what I mean by biodiversity models is the statistical, specifically models that relate species and community composition data. So that includes traits, taxonomic or functional diversity, to environmental and anthropogenic drivers. And this is just an umbrella term that I'm using because the applications that I'm going to illustrate are not necessarily species modeling. So that could be species, but that could be species distribution model, but that could also be macroecological models. So the goal of these biodiversity models is double. The first one is to understand what drives the distribution of biodiversity in general, to attribute this distribution to various ecological processes like environmental filtering, interactions, dispersal, but also to evaluate how sensitive is this biodiversity to the influence of various environmental drivers. The second goal, which is the most focal for this community, I guess, is to forecast whether it's in space or in time and under various scenarios. So in the first part of this presentation, I would like to talk to you about one challenge that is not quite addressed in the classical models, which is accounting for ecological interaction networks as a whole in these models in a manner that allows forecasting. So as you know, species do not exist independently. In fact, they interact either indirectly through a mere shared habitat or competition for resources, or directly through direct ecological interactions like trophic interactions, parasitism, pollination, and so on. And these interactions are typically depicted as ecological networks, as the one that you have illustrated here. And there is, in the literature in the recent years, there's increasing evidence that these interactions influence species distributions. And so accounting for them is important in our models. Moreover, these just not only species, but also these interactions are subject to many to the same pressures that species are subject to. And these different pressures, they jeopardize these ecological interactions that are crucial for species survival. And they do that by and that manifests in different ways, such as by losing co-occurrence between species that are supposedly interact together, such as when two species that supposedly interact respond differently to climate change with asymmetric current shifts, that could lead to a loss of co-occurrence, which would jeopardize this interaction that is between them. Similarly, it can disrupt the phenological cycles that was shown in the study by Böckel in Science, that for plant pollinator networks, but also for migratory birds and their prey. And in most extreme cases, the changes that happen in some compartments of these ecological networks, they can cascade and lead to secondary responses and secondary extinction by propagating through this ecological network. A stark example is when sea ice melts, reducing the range for ice algae, which would provide less food for zooplankton and which would cascade to all the predators or the consumers of these animals. And so now that we agree that we have to account for ecological interactions in biodiversity models, how do we actually achieve that? Especially because the first question that we can ask is, do we know this ecological interaction? And in fact, we are at a ripe time to do that, because in the last years, in the last decade, there's growing interest in gathering and, most importantly, sharing interaction data. There are many databases like Mango, like Globi. We also, in the LECA, we developed a knowledge graph for soil interaction called Trophic Knowledge Graph. And also GBIF is now working on a data model for sharing interactions. And that makes it the best time to actually now more than ever leverage this information. Now, this information comes from different sources. It can be from experiments, observations, from expert knowledge, and sometimes from statistical inference. So to account for these interactions, what I will present to you is a method that was developed by a colleague of mine and Bilio, Giovanni Poggiato. It's a method called WebSDM, and it's an approach that allows the explicit integration of trophic interactions within SDMs. The ingredients for these methods are a set of environmental layers or raster layers, a network of potential interactions. So that's what we refer to as a meta-web. It is typically a directed and acyclic, and that is important for the applicability of this method. And then the third ingredient is obviously in situ observation data for these species. And the idea here in this model is that we fit each species response as a function of the environment and the set of resources on which it depends. That is in the case where we assume bottom-up control by resources for species. And the approach also works on the other way around when we model species as a function of their predators, assuming a top-down control. But the approach cannot accommodate both strategies at the same time because that could create cycles in the graph. So this model makes three assumptions. The first one is that the meta-web, or this network of potential interaction, has to be directed and acyclic. It has to be fully known, and that it is stable in space and time. And it can only account, as I was saying, for either top-down or bottom-up control, but not both of them at the same time. This is the only slide with mathematics. It's here the response, I'm giving it as formulated as a linear model, as a generalized linear model, where the response of the species that we will call mu ig, the mean response of the species, is a linear function of some baseline response, beta GO, the sum of its response to the environmental factors, and then the sum of its response to its prey, or predators, if we assume top-down control. And in fact, for this model and under the previous assumptions, and given this nice theorem called the causal Markov condition, we have a nice property, which is that given its prey, a species is conditionally independent from the prey of its prey. So basically, we can model each species simply as a function of its resources without accounting for other indirect effects. And that is ensured by this property. And so our problem of modeling each species that is structured in a network will translate into S independent local models, S being the number of species, where each species is a function of the environment and its resources. Now, you may say that this is obvious for training the models, and this is actually quite similar to piecewise structural equation models. But the question is, how do we do prediction in new areas where we don't observe the prey of the species that depend on other ones? And in this case, we rely on what is called a topological sorting algorithm. And these are fairly fast algorithms to run for even big food webs. And what they do is that they provide you an order of prediction for the species, such that you predict species subsequently following this order. And this ensures that when you want to predict, let's say here, the top predator, the wolf, you already have predictions for its two prey. Now, one of the potential issues of this approach is that when predators have multiple correlated prey, this can create multicollinearity issues, especially in linear models. This can have consequences, statistical consequences on coefficient estimates. And so to deal with that, the package implements sparse modeling or regularization to reduce model complexity. And this feature ensures that when there is no effect of biotic interactions, then the model will become equivalent to classical SDM that only depends on the environment. Additionally, we may have a situation where we have generalist predators that have an intractable number of prey, specifically omnivorous species. And then in this case, the specific dependence on a specific prey is quite unjustified. We cannot expect that there is a particular strong link with a particular species. So in this case, the package implements a set of composite functions of prey. These are aggregation functions of prey. An example is a function that says, is there at least one prey present? Diversity of prey, whether it's taxonomic, functional, or a clustering of prey based on some functional or trophic clustering that is given a priori. These issues arise both using bottom-up or top-down control. So now I'm going to give you an illustration that we did for this approach, in particularly to predict soil biodiversity in the French Alps. This is a joint work that we did with Giovanni and also with Irene Calderon-Sen, who was at the time a PhD student in LECA, with Olivier François and with Petri. Before I dive into the application, I think it is important to remind that soil biodiversity is one of the most understudied groups, that it is not assessed as much as vertebrates or plants or other more charismatic species. Yet it is a very complex system, and it comprises thousands of species in a given sample, and it is multi-trophic. So in a given small bank of soil, there is so much diversity and so much interaction happening inside. That includes microorganisms, mesoorganisms, microorganisms, and all sorts of organisms that you can see illustrated here. For this case study, I'm using data from the Hors-Champ monitoring program. At that point, where I did this analysis, we had 35 gradients distributed in the French Alps. Each gradient is an altitudinal gradient, as you can see it here illustrated on with the beautiful mountain here. And these plots are separated by altitude level. So every 200-meter altitude, there is a plot that is positioned. And within each plot, we sample within 3 subparts or subplots of this part, soil samples. Now this monitoring program has also points in the Pyrenees Mountains and more recently in Corsica and also in Switzerland. So this might be interesting maybe for your survey of monitoring programs that are potentially useful for forecast. So each of these, then for each of the soil samples that we collect in this plot along altitudinal gradients, we use eDNA metabarcoding with different markers targeting different groups. So we have markers for bacteria, for eukaryotes, for fungi, for columbola, for oligochaetes, and for insects. And then additionally, we also have measures of local conditions, including local temperature and moisture. We have also soil physico-chemical properties, as well as soil types and land use. This information is massive. Each metabarcoding process will produce a clustering of existing sequences for each marker, and each cluster is called a metabarcoding operational taxonomic unit. And each of these units is associated to a taxonomic path. But this taxonomic path can have different granularities in assignment. For some groups like bacteria, we can go up to genus level. For fungi, we can go up to genus level, but for other groups, we sometimes only stop at family level. And so what we do here is, instead of working with sequences or with taxonomic units, we translate these taxonomic units into trophic groups. And to do that, we use this library called Integraph, which is a data integration tool that was developed by Nicolas Le Guillaume in LECA, on which initially integrated interaction data from different sources, as well as data that comes from soil biodiversity experts. And then this data was integrated in a more formal way using soil ontology into a graph called Gratin. We're very attached to the cheese in this area. And Gratin stands for graph of interactions. And so what this process does is that it translates each operational taxonomic unit from a taxonomic assignment to a trophic group assignment. And then for each taxonomic group, we compute for each sample its diversity in terms of diversity of sequences that were assigned to this trophic group. And by trophic group, an example is decomposing bacteria or predator protists, for instance. So these trophic groups have been done in a study by Irene Calderon-Ciano in 2024, and they can be grouped in broad trophic classes, which broadly corresponds to diet or functions, types of trophic functions in the soil. And then these trophic classes are further detailed into trophic groups by combining information from which taxa this comes from. I refer you to this study to see more detail about this trophic assignment. Additionally to that, this carton of knowledge graph has also information about which trophic group interacts with which trophic groups. For instance, bacterivorous fungi would consume bacteria, and then fungi mycoparasites are parasites of other fungi. And then this kind of information is used to reconstruct a trophic or a food web of the soil trophic groups. And this is what we use as our network here. So in summary, we have for each sample, we have the relative diversity of each trophic group. We have a soil metaweb or food web that depicts which trophic group consumes which trophic group. And we also have environmental layers. So we have all the ingredients to use the WebSDM approach. So the question that we asked here is, do trophic interactions explain distribution patterns of soil trophic groups along altitudinal gradients? And is the importance of this trophic interaction homogeneous or heterogeneous across trophic groups? So in terms of modeling framework that we use as environmental data, we use both climate and soil data, but we also use some remote sensing derived variables describing vegetation phenology and also land cover. And here, rather than using a linear model, we implement each species model as an ensemble of machine learning models. And this ensemble of machine learning models are fitted in a spatial block cross-validation setting to avoid overfitting and also overestimation of performances of our models. And this also allows us to have maps of uncertainty related to our predictions. We use different types of algorithms that I will say later. So quickly for the results, the ensemble model, the first result that we had was that the ensemble models were much better than individual models. So it helps to actually use a diverse set of models. In particular here, we used random forest, extreme gradient boosting, and a support vector regressor. And we found that the ensemble approach actually outperformed these in the individual models. On the right, what you have is a graph that shows the difference between a baseline model that only uses the environmental information and does not incorporate the trophic information. So basically a classic ensemble SDM minus the difference to the trophic SDM. So what you see here is the difference trophic SDM minus the classical SDM. And as you can see, the differences in performances here, the metric is Spearman rank correlation. The difference in performance here is mostly positive for pretty much all of the groups, except a few exceptions that are actually not well modeled. But for most of the groups, we have a great increase in performance when adopting this approach. There is particularly the one group that benefits a lot from using this approach. It is the one that with the big blue purple bar. This is the group of mycoparasite fungi, and these are fungi that only exist with the existence of their host, which are other fungi. And so that makes sense that the environment would never explain their distribution alone. Next, what we try to look at is the relative importance of different groups of environmental variables and the resources. Here in red is the importance of the trophic resources or the prey. And we see that this importance varies across taxa, so it is quite heterogeneous. And this biggest contribution is for fungi. Again, this is due to this group of mycoparasites, but it was also quite important for columbola and also from some predator insects. So then we used the approach for forecasting using this topological ordering algorithm. And we produced maps for each of these trophic groups. We have examples here. For instance, you have the ectomycorrhizal fungi in the center. And then additionally, we looked at composition to see if we can have if you can predict clusters of trophic groups, clusters in the Alps of regions with the same trophic composition in terms of trophic groups. And this is the map that you can see on the left here. So I will not dive deep into the spatial results, but what I want to take home as lessons is that this framework is quite flexible. It can incorporate ecological interactions as shown. Its use cases are not only for better understanding what drives species distribution, but it was actually shown to improve niche estimation in simulated settings, obviously. But it can also be used for forecasting. Here I've shown you an example of forecasting in space under current condition based on past year's data. It can also be used to quantify the strength of interactions because we usually can get information about who interacts with who, but into how much dependence this translates and its effect on occurrences. This is thankfully possible through this framework. It can also allow to propagate uncertainty because there is an implementation in the package that uses a Bayesian framework to ensure that the uncertainty from predicting prey can propagate to the uncertainty of predicting the predators that depend on them. In general, this is applicable to all problems where the system is described as a directed acyclic graph of its elements, not just species, but we only tested it on species. Now there are some issues related that still remain with this model. The first one is that these metamaps, they need to have this directed acyclic graph structure. One solution is to couple this kind of model with joint species distribution models or other types of graphical models for the type of interactions or associations that are undirected and symmetric, whereas interactions that are more directed, like trophic interactions, then in this case, you can use the approach that is proposed here. There is a lack of uncertainty on the potential interactions. We assume that we know them, that we are sure of them. One way to address this is to adopt a Bayesian framework and to fit the model with different potential networks. A third limit is that these interactions are assumed to be fixed in space and time, and that they are not context dependent, which there is evidence that it is not always the case. So in theory, this is feasible, but that violates some of the assumptions that the model is using. The last, but maybe the most crucial part is that it is quite difficult to have a complete network of interactions for all the species that are sampled, especially when you're using environmental DNA and you are very exhaustive in what you're observing. In this case, it is possible to use information on interaction at a coarser granularity, like we did with trophic groups, or to use some statistical methods to predict missing links in the network. This was done before by approaches like trait matching in a study by Bischler and al. In 2020. And the last idea that is maybe the most in tune with the latest advances is to use these large language models and retrieval augmented generation tools to extract the existing information that is in the scientific literature, but that is not on the databases. So in practice, for forecasting, just a few lessons, or a few, let's say, a recipe for using this model for forecasting. The first step is to choose a direction of interaction. In its current form, the framework only supports either top-down or bottom-up control. To use composite variables to prevent this multicollinearity issue, and also to avoid overfitting a predictor model to a specific prey. To also to follow this topological order technique to predict with the model, and to choose an appropriate species model that depending on your data. So the model, how we tested it, was mostly on static data, but it can easily be tested on temporal and time series data. And the most important part is to propagate uncertainty by either using a Bayesian approach, and that is already implemented in WebSDM, or by using a more frequentist approach where you generate multiple samples and you aggregate them statistically. So that's it for this part. Can either take questions now, or I can continue and take all the questions at the end.
We can do it at the end, I think. So no worries.
That's good. So for the second part of this talk, I would like now to focus on mostly on the environmental part, on how we can use AI to enrich the set of environmental predictors that we use. So the motivation for this is that when we look at the drivers of biodiversity change, the most important one is habitat loss. And habitat is itself responding to climate and other sources of pressures. So there is a need to model habitats themselves continuously. And so in addition to these species distribution models that I talked about in the macroecological models, now we are more and more working on these habitat distribution models where we model the response of habitat types or vegetation plant communities as a whole to environmental change. And these kind of models, they give you estimates of habitat extent and also habitat condition. There's now a nice opportunity with this massive Earth observation data that has been collected over the last decades. And this image is only for the European fleets. There are also many existing missions from NASA and from Japanese missions and so on. Typically, what is done with this kind of data, it comes in the form of time series of images that cover a large spectrum from visible bands like red, green, and blue to infrared and to shortwave infrared. And so there is it's three-dimensional in a sense that it has the spatial dimension, the spectral dimension, and the temporal dimension. What is typically done is that this information is summarized along the spectral dimension into various vegetation indices. An example is the NDVI, or Normalized Difference Vegetation Index, or the Chlorophyll Content Index, which is a summary of that combines different spectral bands into a meaningful interpretable index. And then these indices are analyzed over time. They're dynamic over time, particularly during the growing season, but not necessarily, are analyzed to extract indicators of vegetation phenology or productivity. They can also be used to analyze vegetation structure parameters, such as leaf area index or canopy density. And so this, or they can also be used to classify into land cover maps. So this gives us a set of what is called remote sensing biodiversity products. They are a proxy to the habitat condition in a sense at a given time or over a given period. But typically this information is very high resolution, and sometimes the sampling areas can be bigger than actually the resolution of these images. And there are many reasons why taking the local information from this related to our sampling areas is not sufficient, and sometimes we have to take information at the landscape level. One of the reasons can be purely technical, related to data challenges such as location uncertainty. We have an example here of an observation of a habitat where you have in red the location is in the center of this image, but actually the sampled habitat is a grassland, and the grassland is mostly is not on that location. So if you take the remote sensing data on exactly that location, then it is likely that we will average phenology or vegetation structure parameters that are incompatible with grassland ecosystem. Another motivation is that sometimes we need to match the sampling area. For example, here with the hors-champ monitoring programs, typically the monitoring plot is 30 meter by 30 meter. And here on the left, what you have is an aerial photography. The aerial photography products that are available in France are at 20 centimeter resolution. So you cannot just take the 20 centimeter in the center of the plot, which is just a small part of the area that is sampled. And so here, it's the whole plot information, the landscape level information that needs to be summarized. Another motivation, which is probably the most important, is the ecological relevance. Sometimes the important factors that drive the biodiversity distribution are not necessarily local, but the observed species are also subject to pressures or to processes that occur at landscape level. For instance, if you take a pollinator, it's important that its habitat is connected to the habitats where it goes to perform its function of pollination. Similarly, some biodiversity can be subject to some geological processes or geomorphological processes like erosion that actually occur on a larger extent than the area where we're sampling. And lastly, some species have a very large home range, and it doesn't make sense to model them at only the locations where they are observed, because that can be any location within their home ranges. So the question is, how do we best summarize remote sensing data at the landscape level? Maybe the most obvious way to do this is to simply compute some statistical summary or landscape metrics based on the remote sensing biodiversity products, like the landscape composition metrics or average, minimum, maximum values of these different products. Another way to do this is to look at these raw images and to analyze them using computer vision algorithms. And this is what I would like to talk about now. So typically, the state-of-the-art computer vision algorithms are mostly based on deep neural networks, and particularly two types of architectures, so convolutional neural networks and vision transformers. And what they actually do is they take an image as an input and they transform it into a new vector of image features describing the information within this image. You can sometimes hear these concepts of embeddings or vector representations, which refer to this set of image features. How are they concretely learned? They can be learned in different ways. Either what is called the end-to-end training, and by that you mean if they are trained, if you fit a model directly on your problem of interest. But in this case, it requires large databases and it has a very high carbon footprint. And depending on the architecture, it can require even more data. For instance, vision transformers require much more data than convolutional neural networks. So this approach is now probably deprecated, and not many people do that now. Instead, what is typically done is to use transfer learning. And what transfer learning is a process where you take a model that is already somehow pre-trained on a similar task, and then you fine-tune it, or you try to specialize it on your problem of interest. For instance, if I want to model habitats, I can take a model that already was trained on land cover. Land cover and habitat is a similar task. Similarly, if I take a model that was trained on vegetation cover, then it can be relevant for underground, below ground biodiversity, because it can it's somehow related, or at least they can share different drivers. But this still requires a bit of data to do this fine tuning, less than this end-to-end training or the from scratch training, but at least it requires less data. The third approach is what is called self-supervised pre-training, and here these are models that are referred to as foundation models, and that are typically trained without any supervision. So these are models that are not trained on a particular task. Instead, they simply learn to extract these image features by solving some pretext tests. An example is an image that is one of the examples that is used is they take an image, they make some holes in it, and then they try to reconstruct these holes. And this process creates a model that learns generic features and that is not specialized on a particular task. And for Earth observations, there was a surge in the last maybe 5 years of these foundation models for Earth observations. So what are these Earth observation foundation models? These are deep learning architectures that are pre-trained using this self-supervision on large multimodal Earth observation datasets. By multimodal, we mean different sources of Earth observation, including both multispectral, hyperspectral, radar, LiDAR, etc. How do they work? They learn general habitat landscape features across spatial and spectral dimension using this process of self-supervision. And you can see here the example I was describing earlier, where you have an image where parts of it are hidden, and the model is trying to predict or reconstruct the missing parts. And by this process, it learns to describe the image in a way that allows it to reconstruct if there is a missing information. Their usefulness is mostly in saving training time and carbon footprint, but they can also be quite useful in problems where you have where you are very scarce in data. For instance, collecting data on soil biodiversity is quite time consuming. Maybe not as costly as before, but at least it's very time consuming, and it requires going to the field. And so the few data that we have is very precious. And so in these situations, you might want to have a model that does not require that many data. And so I will go quickly to this application where we use these foundation models to map habitats across Europe. And this was a collaboration work with Stefan Hannikens and Sander Lucher from the Wageningen University. The reason why we worked on habitat is because these are one of the components of conservation concern in Europe. They're almost 75% of them have been assessed in poor or bad state, and it is important to know where we can find where these habitats that are in bad state are located, and this is important to both inform the European biodiversity strategies, but also to inform also restoration programs where we want to know areas where the habitat could be, but where they aren't actually. And so in practice, we use this habitat classification that is called UNIS, and which defines habitat as association of vegetation and their environment. It is broadly grouped into 9 categories at level 1, but it can go up to level 5. For this study, we stopped at level 3, which is already 257 classes that need to be modeled. And this includes marine habitats, coastal habitats, freshwater, wetlands, grassland, forest, and so on. As a data set, we use data from the European Vegetation Archive over Europe, and we also used some independent data set from the Netherlands, Austria, Portugal, and France to validate the models. Here, the question that we asked was, what is the best strategy to model habitats at European scale, and how can remote sensing and AI contribute to that? As environmental variables, we used a set of abiotic conditions, including climate, soil, terrain, and hydrography. We coupled that with remote sensing biodiversity projects as the most richest set that we could get. And then we wanted to see if by adding these remote sensing images in their raw format, whether they would bring information or not. And in particular, we included multispectral information from Sentinel-2 and also radar information from Sentinel-1. And you can see some examples here. We tested images of different size from 250 meters to 1 kilometer. And then we use these Earth observation foundation models to translate each of these images, so the multispectral and the radar images, into these features or embeddings. And this provides us with a new set of features that describe the landscape more from the raw format, and they are not that interpretable, but at least we can try and test if they have any, if they provide any additional information. And so we tested models with increasing complexity of data. We started with a model that only used the abiotic information. Then we included the remote sensing biodiversity product. Then we included the multispectral information, and then we included the radar information. Each of these models is an ensemble of machine learning models, and it was trained using the spatial block cross-validation strategy to provide both ensemble mean averages and also uncertainty. And then the ensemble mean averages habitat maps are validated against this validation dataset. So quickly for the results, what you can see here in blue is the results with the model that only used no remote sensing. And we can see that we already have good performances for some habitats and average for others. When we add the remote sensing biodiversity product, which was a quite rich set, we increased considerably the performances, particularly for non-vegetated habitats and wetlands. And then when we add the information from the imagery, we actually increase considerably, both in terms of recall. So recall is whether we detect the true class that was observed, whether predicted as the most likely, and precision is whether we are predicting spurious habitats. And we can see that adding the images actually improves on both dimensions across all habitat types. Just a quick note on these Earth observation foundation models. We compared an approach where we fitted a model from scratch, end to end. This is what I call here the EO4B model that is highlighted in red. And this is a model that was trained from zero using the data that we have, which was already quite a big data set. And what we found was that all the other approaches that relied on these Earth observation foundation models actually outperformed it, even though they were not necessarily pre-trained on this task. But these Earth observation foundation models, amongst them, there was no systematic ranking, and it was quite variable across datasets. So then we used this model to predict habitats across Europe. First, we used the models for each broad habitat type to predict the most likely class. And then we applied a set of filtering by regional, by ecoregions to account for endemisms, and by land use to produce a map of European habitats at European scale. And so in summary, what I'd like you to take from this part is that remote sensing adds unique ecological signal. Although these abiotic products, they were most informative, were not insufficient alone. The RS biodiversity layers, such as phenology and canopy height, were critical for fine distinctions of habitats, and they were complemented by the other sources of data. these Earth observation foundation models consistently outperformed the models, the supervised models that were trained from scratch. And we actually interestingly find that the size of the image matters. We find that using larger images for forest and wetlands was better, whereas smaller images for mosaics or grassland was more appropriate. And then maybe in terms of limitation, we use these Earth observation foundation models as they are. We did not further specialize them on our task. And this is one potential extension or one potential way to improve the models. The data that we trained on is point data, so it's not polygons, and so it does not delimit the habitat itself. So it was quite challenging to find or to model the extent of the habitat with this kind of data. And finally, the sampling density was uneven across Europe, and that can reflect in the model predictions. So to end, this kind of landscape descriptors that we derived from these Earth observation foundation models, they provide these generic high-dimensional embeddings that summarize the information across the spectral, the spatial, and maybe even the temporal dimension across large areas. They can be computed over time with satellite time series to enable near casting. And so they can be used also as dynamic covariates, and they are transferable in new areas. So, but it is still limiting for future forecasting in more decadal timescales, where it is quite impossible to invent or at least to guess the remote sensing imagery. Practical consideration that you need to keep in mind is that the resolution and the process of the Earth observation foundation model has to match the ecological process that you're trying to account for. The temporal frequency of this Earth observation data is important to keep in mind, and to always integrate this other interpretable remote sensing product to the analysis. And then in terms of computational tractability, these models can be quite big and huge to predict over a continental global scale. But thankfully, there was some new developments in Earth observation foundation models where they are now produce raster layers of these image features, so that you don't have to go through this heavy process of predicting these models. And so just to end this presentation, what I went through in this presentation was we started from this biodiversity models that relate biodiversity data to environmental data. We first saw an approach to account for dependencies between species, and then a new set of environmental features to enrich these models by describing landscape structure. Yeah, that's it. I would like to acknowledge my nice collaborators and also the funders, Miai, Natura Connect, Obsession, and also data providers. And many thanks to the amazing team that we have at LECA. And thank you for your attention. I'm happy to answer any questions.
Thank you very much for this lovely talk. And it was really, really interesting. And I think all of us agree. And yeah, sorry again for going a bit over time because of our connection issues. But there's still some time for questions. And people were also asked who had to leave earlier to ask questions in the chat. But I think there was no questions in the chat to be seen. So are there any questions from the live audience? While people, there we go, Owen, good, because I was about to ask one. Go ahead.
Thanks, Sarah, for that very nice presentation. I'm particularly interested in the first part because I have done some modeling of food web structure, predicting interactions predicting food web interactions So I guess this isn't actually a question, it's just a shout, it's just a maybe something that we could talk about more, and when I could describe the work that I've done a bit more on predicting interactions, particularly including dependence on environment and dependence on other species, presence, absence. So it's a kind of model that doesn't make this assumption that there's stability in the metaweb. There's no metaweb at all, really. It's just a model of the interactions, like the trait matching, but a bit different. But yeah, really very nice. Actually, I will ask a question as well. Why don't I ask a question properly? It's a relatively straightforward one. You concluded with, there was a slide, I've taken a screenshot, so that's not allowed, on the ensemble performance of the food web model, of the web SDM. Actually, the more interesting question is, like, actually, can you help me understand what these absolute measures of what these difference values mean? Like, in terms of how much better the models are. You know, I'm seeing the F mycoparasite up above 0.25. What does that
I can maybe go back to the slide to have it. This one, right?
Right. What's that? What's some kind of intuitive understanding of that x-axis on the right-hand graph?
Okay, so maybe to have an intuition of that, I should explain the metric on the left-hand graph. So the left-hand graph shows the spin run rank correlation, and what it shows is whether we predict the species as, if you take two sites, and in one site it is more abundant than on the other site, then the model should at least predict well that in the first site it's more abundant than the second site. So it keeps at least the ranking of sites. Because here we're not necessarily interested in the absolute value, but rather in whether in the ranking of locations. And then on the right-hand side is the difference of this metric between the models that uses basically the trophic SDM, the web SDM, and a model that simply just uses the environment with this exactly the same setting, so the same data, the same environmental predictors, but no accounting of resources. And this is the difference between the web SDM minus the classical SDM.
Thank you.
Is that clear? Yeah.
Yes.
Also, I would be very much interested by the ref to the work that you've done on predicting interactions.
Sure, I'll put it, I'll couple in the chat.
Thanks.
Yeah. Era. Thank you, Owen.
Yeah. Yeah, thank you so much for this talk. It was really interesting. I was also curious about this part, and with the web SDM stuff, with the assumption that the network is either bottom up or top down controlled. And I was wondering, like, one, if you think that this is something that can be modified in the future to have either like a mix of, you know, both control mechanisms going on, or also like some species being more influenced by bottom up and others more top down. And then two, in the current state where you have to kind of assume one, can you test like which one performs better? Is that kind of part of the workflow with this approach?
Yeah, you can test both of them and see which one explains already the biodiversity patterns more than the other. That's obviously possible. As extensions, I think if you use dynamic data, you can mix top-down and bottom-up control. It's mostly if you use in static data, because in this case, it can create cycles. If you're on one part of the network, you're using top-down, on the other one, you're using bottom-up. But if you can compartmentalize it into components of the network, in which for one of them, there is only dependence within this compartment and it's top-down. And then for another compartment, it's bottom-up. Then it could work. But more majorly, the biggest issue is this cyclicity, because to compute the topological order, we need acyclicity. Yeah.
That makes sense. Okay, cool. Thank you.
Yeah.
Thank you, Kara and Biller.
Thank you so much, Sarah, again for the great talk. I was wondering about this for the prediction side of the WebSTM for this topological sorting part. So basically, the idea of using the prediction of one occurrence data for predicting the other one. I was wondering if there is an issue of almost double counting of the climate there, because you use the same climate to predict one of them and then you use it as again. I think you somehow there is a regularization part in the package, and I was wondering if that also somehow helps with that kind of double counting of the climate.
It seems that way, that there is double counting of climate, but if you assume that all your species depend still on the climate as a direct effect, then in this case you're actually accounting for both the direct effect of climate and also the indirect effect through the price. And so it's important to account for it in both models, for the resource and for the consumer, because you want to separate the direct and the indirect effects.
Yeah.
But maybe I agree. Maybe for the prey model, maybe it's also capturing somehow part of the response of the predator. It's still all correlative models after all.
Yeah.
Yeah, but I think you still need to account for climate for all species to make sure you don't miss out on the abiotic part, which is mostly the most important.
Yeah. Thank you.